according to Francis Crick, who, with James Watson, won the Nobel Prize for their discovery of the three-dimensional structure of DNA, I myself was forced to call myself a molecular biologist because when inquiring clergymen asked me what I did, I got tired of explaining that I was a mixture of crystallographer, biophysicist, biochemist, and geneticist, an explanation which in any case they found too hard to grasp. Indeed, in our study of cell and molecular biology, we are required to look back at our previous knowledge on chemistry, physics, and biology itself. And I believe many of you right now are taking courses such as biophysics, biochemistry, and genetics in addition to this course. So we need to be recalling and connecting the knowledge and the concepts that we learn in this course with these other courses. But first, let's look back at the history of the cell and man. How did man first encounter the cell? We are very much aware that it was Robert Hooke who was first to see the cell under the microscope. Even though it was a non-living cell, he still beat everyone to the record. So Robert Hooke was fortunate enough to have access to the microscope during his work. And the microscope during his time was a prized possession, new and very current to the scientific community. So because Robert Hooke had access to the microscope, he had the luxury of observing many things under the microscope. One object he observed was a thin slice of the cork tree's bark. And this is what he observed. So he drew them, and he observed that the bark of the cork tree is made up of squares. Squares that seemingly look like the rooms in an abbey where monks lived. So because these squares reminded him of those cells, he coined the term cells to call these things. But the encounter of the cell by man didn't stop there. Very shortly after Hooke's story, Anton van Leeuwenhoek, a Dutch scientist, was the first person to view live cells under the microscope. But his story of the use of the microscope didn't come easy. Leeuwenhoek was unlike Hooke, where he didn't have access to the current technology, such as the microscope, during his time. Instead, Leeuwenhoek made his own models of microscopes from scrap. So we can see here a picture of Leeuwenhoek's microscope. This is one of his models, and it looks very different from our view of what a microscope is. So there is a lens, which is hardly covering a hole on a metal plate. And then there is a screw-like mechanism, and at the tip of the screw, the specimen is going to be placed to be viewed under the lens. So Leeuwenhoek would often bring this microscope near his eye, squint, and look through the lens to the specimen. And he observed, as I mentioned a while ago, the first living cells that were seen by man. So including cells from the build-up on his teeth from blood and from pond water. And he also drew them. So in his book, you would often see these squiggly lines, as shown here, which depicted the shapes of the cells that he saw. And often he would be drawing arrows indicating that they were moving from one point to another. And because they reminded him of animals, albeit they were really, really microscopic, he called them animalcules. Many years later, when scientists and people have become very much aware of the presence of cells, the cell theory was born. But it didn't always stay as it is, as we know it today. Two scientists, a botanist, Matthias Schleiden, and a zoologist, Theodor Schwann, actually looked at specimens of both plants and animals, countless of them, under the microscope. And they all agreed that all living things, regardless of being a plant or an animal, is made up of cells. So they agreed also that this cell is the structural unit of life. But believe it or not, at this point, even these two scientists still thought that life can come from non-living things. So the theory of spontaneous generation was still popular and held correct during this time. It was later on, with a contribution of Rudolf Virchow, wherein he said that living things or cells may only come from pre-existing living cells. So Virchow was a pathologist and a microbiologist, and it's very likely that he observed bacteria giving rise to new bacteria under the microscope. So today, we have the modern cell theory comprised of these three statements. Arguably, the third one, which says that life can only come from life, is the most important among the statements. So, I encourage you to read further chapter 1 from our book, but I will be stopping my introduction at this particular point. Instead, I'll be leaving you with some questions that you should be able to answer once you've finished reading chapter 1. So first, what is reductionism in the context of cell and molecular biology? And what are its advantages and disadvantages? Second, is there an alternative to reductionism? And if there is, describe it briefly. Third, why do you think the idea that only living things can give rise to living things came very much later than the idea that all living things are made of cells? And lastly, there are smaller building structures of living things, even smaller than cells, such as atoms and molecules. Why is it then that the cell was chosen to be the basic structure of life and not the atom or the molecule? And support your answer.